Good morning, students. Today we will study about oogenesis. It is the formation and maturation of ovum. It takes place in Graafian follicle. Oogenesis is initiated in embryonic stage when millions of egg mother cell oogonia are found within each ovary. No more oogonia are formed and after added after birth. Oogonia multiply to form primary oocytes. They enter into prophase 1 of the meiosis and get temporarily arrested at that stage. You can understand it with the help of diagram. This is corpus luteum. Ovulation from here. Uterine tube, that is secondary oocyte. Zona pellucida, first polar body. Corona radiata. Each primary oocyte gets surrounded by a layer of granulosa cell to form primary follicles. A large number of primary follicles degenerate during the phase from birth to puberty. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovary. Development of ovarian follicles. Primary follicles, that is your granulosa cell, primary oocyte layer of granulosa cells surrounded by a primary oocyte. Second case, secondary follicles. Theca cell, that outermost cortical granulosa cell, zona pendula, more layer of granulosa cell and a new theca surrounded a primary oocyte. Tertiary follicle, atrium, theca interna, theca externa, granulosa cell. Fluid cavity filled develop theca layer form inner theca interna and outer theca interna. Now for last is Graafian follicle, fully mature follicles. Now theca externa, theca interna, antrum, glossa cell, corona radiata, that you can see. Last is cumulus operus. Primary oocyte within the tertiary follicles grow in size and undergo first unequal mitotic division to form a large haploid secondary oocyte and a tiny first polar body. The secondary oocyte retains the nutrient-rich cytoplasm of primary oocyte. It is unknown that whether the first polar divides further or degenerate. The tertiary follicle further changes into mature follicles. Secondary oocyte form a new membrane, that is zona pellucida. Graafian follicles now rupture to release the secondary oocyte, that is ovum, from the ovary. This is called ovulation. Graafian follicles now rupture to release the secondary oocyte, that is ovum, from the ovary. This is known as ovulation. The diagram, you can understand this. Here, first is gonia 2n haploid, that is 46 chromosome. That mitosis embryonic stage, the primary oocyte of 2n will develop. That is grow in size, and the primary oocyte of meiosis will take place prior to ovulation. Now the secondary oocyte, that is N haploid first polar body. During fertilization, that ova is again in the secondary polar body. Structure of ova: spherical and non-motile, size is about 0.2 millimeter in diameter. Ovum has three membrane: plasma membrane, that is layer, innermost layer; zona pellucida, that is outer to the plasma membrane; corona radiata, outer layer found by follicle cell, that we can see with the help of diagram. This is plasma membrane, inner layer, plasma membrane. Second is zona pellucida, here, this layer, second layer. Corona radiata, this, that is your corona radiata, and that in between, that is cytoplasm, this is the nucleus. This is the difference between spermatogenesis and oogenesis. Spermatogenesis occur in testes, whereas oogenesis occur in ovary. Unlimited growth phase is there in spermatogenesis, elaborated growth phase in oogenesis. Each primary spermatocytes give 4 sperm. Each primary oocytes give only 1 ovum. No polar body formation. Polar body are formed, begins at puberty and extend up to senility. Begins of the embryonic stage but suspends up to the puberty. It ceases around the age of 50. Thank you for today.